next pitch is on tackling standardization issues, and I think we have heard, or according to your votes, this is really an important topic, and in MICROBE we have a dedicated work package on standardization and quality control. And why is it an important topic? I think we all know that there are a lot of factors that can influence a testing result, and this can be at the pre-analytical phase, at the analytical phase, at the post-analytical phase. But I think you all know that the pre-analytical phase is one of the most important ones, where in the human diagnostic field, in human healthcare, it causes most mistakes and can lead to patient harm and even wrong diagnosis. But also in research, we see that there is a lot of money wasted, their results are irreproducible or not comparable, and this is why we saw questions arise like, how does science go wrong, or do we have a reproducibility crisis, that most of the researchers in a survey answered with yes. And this is not only in the human field, but also in the microbiome field. The pre-analytical phase is very critical, but it is still often overlooked or neglected or underestimated. And that's why we put this focus also on it in MICROBE. Now, the human situation, the human field has reacted to this shortcoming, and we have here the in vitro diagnostic regulation, the law that requires now the manufacturers to test and describe some pre-analytic parameters for their products. We have several accreditation standards for the diagnostic field, but also for biobanking. And we have dedicated pre-analytical quality standards from CEN and ISO that were produced or generated in SPEDIA 4B, also an EU project where we from BBMRI contributed. And one of these pre-analytical standards, CEN TS 17626, focuses on human specimens intended for microbiome analysis. And this is a European standard at the moment, but it is currently developed to an ISO standard, 18701, that might be published in 2 or 3 years. And this standard covers the entire pre-analytical phase. It starts with the donor sample collection and goes up to DNA analysis and storage. And here it defines or highlights pre-analytical variables and gives recommendation and requirements what to do and what to document, so that the users then have a support in defining which technical process requirements and metadata requirements are relevant for their projects. So in MICROBE, we took this human standard as a template and transferred it to the non-human samples like soil, marine water and plant, as shown here. And this is one of the approaches we took to overcome this kind of silos, to use the same standards or orientate on the human field. And comparable to the human situation, you see the pre-analytical workflow we defined and the variables we identified. We have a lot of steps, we have a lot of variables at each phase, and all these can impact the microbiome data in the end, because it can modify the sample, the microbiome profile, the samples die or proliferate, the DNA can degrade and so on, and the biomolecules can also be modified. So I will not go into detail, but I can invite you to watch the posters we have in 3A and 8C on this topic. But what I can say is that in the non-human field, the term pre-analytics is not really established, but still there is also sound evidence from literature that some pre-analytical variables have a drastic effect. For example, microbiome DNA extraction protocols, we know this from the human field. Here plants were tested with 11 different microbiome methods and extraction protocols, and the result shows that they are quite different, the outcome. And the reason is that we have different homogenization methods, we have different lysis efficiencies for the plant cells, but also the microbiome cells. There are different amounts of co-isolated inhibitors and host DNA, and also some of the kits or regions, they also contain microbiome DNA from the production phase, so kind of contamination. We also know that or see that the sampling time and the sampling site influence the outcome and lead to different microbiome communities. So on the left side we have leaves from the same apple trees collected in May versus July, August, and you see the microbiome community difference. The clusters are according to the sampling time. And on the right side, flower and leaf samples from the same type of apple trees. They cluster according to the site where they were collected. So strong influence also from these effects, and even the sampling process, for example, homogenization before or after subsampling can influence and lead to different results with only very little overlap. So we really see that these factors, the pre-analytic factors, matter also in the non-human phase. The critical thing is that these pre-analytic effects remain often unnoticed or are invisible until we want to reproduce results or until we run multi-center studies. Here an example from the human field from BBMRI LPC, where different biobanks collected and processed human serum samples, and one side performed metabolomics. And you see the result clusters according to biobanks and not not by patient. So in other words, the intercenter variation is significantly greater than the intra-individual variation. And one of the reasons is that what we do in practice often differs from the very simplified and ideal workflows we show here or that are also in the standards, because the reality is often more complex. Usually we have several intermediate storage phases. The samples might not be preserved immediately after collection, but later, at a later time point in the lab. So this increases also the phases of storage and the time to preservation that is very critical. Sometimes the sample might be even shipped to other partners, as it was the case in our MICROBE project, because we wanted to test different preservation methods. So this was done by different partners. But there are other situations maybe where the samples are immediately analyzed after collection. So you see, in reality, this is much more complex. So we do not only have multiple steps with a lot of variables, but the steps can be in different orders. We usually have many different intermediate storage phases, in addition to different methods, to different procedures, regions. Even if the regions are the same, they can be older or not so old. It can also make a difference. And we have the operator variability. And this in the pre-analytic phase, but also in the analysis phase. So that's why we really see a clear need for harmonization or even standardization of the workflows, but also for documentation and transparency about these procedures. In MICROBE, we made a similar observation. So in the non-human field, it's not different. We had homogeneous aliquots of one soil sample, and this was sent to different partners, and the different partners performed multiple or different kinds of preservation methods and durations. And then they used the same community analysis method with the same primers, and also here the results clustered by partners and not by the preservation method. So also here the intra-laboratory variation is a lot larger than the preservation effects we see. This means also that just a rough alignment or harmonization of the methods might not be enough, but sometimes it really needs shared SOPs. And this is actually what we then did in MICROBE. We sat together, we standardized all the workflows, the pre-analytic and analytical workflows, and we wrote SOPs, really one SOP that was used by all. This was done together with the entire lab team. And still we see some situations where not every pre-analytic variable can be controlled. Here, like for example, the shipping duration and the shipping temperature. here samples from same sample types were identically packed and sent off the same day, and one package arrived a day later, and the temperature curves, they differ. At least we have documented the data, so this is good. We have taken the measure, we have documented it, and we can, in the end, use this to explain our results a bit better when there is, for example, an outlier. So if we want to have high quality data, microbiome data, and also fair microbiome data, then we need high quality samples, and of course we need the important metadata which are often not collected, and therefore we need these quality control workflows that tell us which pre-analytical process and metadata requirements are there. And this will then help to capture the microbiome as it is in situ, and not that is a result of the artificial pre-analytical handling. It shall increase, or it will help to increase reproducibility, and also is important for the secondary use or reuse of samples and data, because then if the procedures are reported, then you know the sample history, how it was treated. And this then can also be useful for a researcher to find whether the sample is fit for purpose. So with this, I'm at the end. I just want to highlight we also developed a guidance document for sample pre-analytics for the plant field that is available online. And I also want to thank all the colleagues who contributed to this work.